Hello everyone, and welcome back to VetCare. Today, we're exploring one of the most fascinating and essential topics in veterinary science, the sequence of reproductive events, along with a brief journey through the history of reproductive physiology. You can think of the sequence of reproductive events as the backbone of reproductive physiology, the natural step-by-step journey of how reproduction happens in animals. It all begins with prenatal development, when the reproductive organs are formed before birth, and continues to puberty, the stage where animals become sexually mature. Once maturity is reached, the reproductive tract is functional and ready for gamete transport and fertilization, with the entire process regulated by key hormones such as GnRH, FSH, LH, estrogen, progesterone, and testosterone. In females, reproduction follows cyclicity, with repeated estrous cycles, while in males, spermatogenesis ensures continuous sperm production. Following this, ovulation occurs in females, leading to copulation or mating, where semen is deposited into the female reproductive tract, and fertilization takes place as sperm and egg unite to form a zygote. This is followed by early embryogenesis and maternal recognition, where the embryo signals the mother's body to maintain pregnancy. Afterward, fetal attachment, gestation, and placental development ensure proper fetal growth until pregnancy concludes with parturition, the process of giving birth. Finally, the puerperium period allows the uterus to return to normal while lactation begins to nourish the newborn. While this is the natural sequence, it took centuries for humans to uncover these secrets. Aristotle, as early as 384 to 322 BC, believed the fetus originated from menstrual blood. Much later, in 1562, Fallopius described the fallopian tube, followed by Coiter in 1573, who identified the corpus luteum. In 1672, Graaf described the Graafian follicle, and in 1677, van Leeuwenhoek observed sperm under the microscope, calling them animalcules. By 1780, Spallanzani hypothesized that sperm were the true fertilizing agents and even performed the first artificial insemination in a dog, while in 1825, Dumas confirmed sperm as the actual fertilizing agents. From there, science entered the modern era of reproductive physiology, which paved the way for groundbreaking techniques such as in vitro fertilization, estrous synchronization, embryo culture and transfer, and frozen semen technology, all of which have revolutionized both veterinary and human medicine. And that brings us to the end of today's video. If you found this video helpful, don't forget to like, share, and subscribe to VetCare for more simple, clear, and exam-oriented veterinary lectures.